known as IOPAN. And the Oceania goes to the Svalbard archipelago every summer to do some long-term monitoring of the physical and biological environment. And I was able to join the cruise in the summer of 2018 to collect some specimens. Now, coming to the project, Molecules Meet Fossils. So we know from the fossil record that diversity has fluctuated through time. And this is the famous Sepkoski curve of marine invertebrate families over the last 500 million years. And you can see that there are peaks and troughs of diversity, where the troughs are indicating the mass extinction events. And what we're interested in is to look at in a bit more detail why and how this diversity fluctuated through time. We're doing this by integrating molecular evidence in terms of molecular phylogenies and the fossil record. Now, our study group are bryozoans. These are colonial aquatic invertebrates. They live in marine and freshwater environments, all the way from the intertidal to the deep sea, from the poles to the tropics. And most of them produce a calcium carbonate skeleton. And that means they fossilize really well. So we've got a really good fossil record with about 15,000 fossil species currently known. In terms of their living diversity, there's about 6,000 species described, but as we are collecting, more and more species, new species are constantly being discovered. So this is a huge underestimate. In terms of their colony forms, so we can have encrusters like here, the bivalve encrusting species here. We can also have hard erect colonies like these guys here, or we can have more arborescent colony forms like miniature trees with articulated joints. So these are flexible branches. So this is only some of the examples of how a bryozoan colony might look like. And many of them are actually quite cryptic, quite small. In terms of the colony organization, so the colonies are composed of individuals called zooids, and in bryozoans, the zooids can take on different forms and functions, so they're highly polymorphic. So this is an example of two different species, and so we can see here, this would be a feeding zooid, so the tentacle crown, the filter feeding tentacle crown would come out here. So that would be for feeding, function of feeding. Then just below it, we have a zooid that is responsible for defense. So you have this mandible here, this gray triangle, which flips open and shut, pivoting on this pivot bar. So the mandible is actually gone in this case. And it stops predation and anything settling on the colony. And then we also have reproductive zooids. So here, So here we have this is what's called an ovicell. This is a calcified chamber which incubates the embryos. So this is being produced as we speak. So this is a partially formed ovicell. Whereas if we look at this species, we can actually see fully formed ovicells. So this is where the embryos are being brooded. Here we also have defensive zooids. Here's one, and they have a different type of defensive zooids, which are here on the sides of the branches, and they have a very, very elongated mandible, and these are almost like whip-like structures that whip back and forth to prevent anything from settling on the colony. In terms of our project, so we're interested in diversification dynamics. And here at the top, we have the ingredients that go into our analyses. So we have innovations, so functional traits, morphological traits that may have led to an increase in diversification rates. Then we have a molecular matrix of DNA sequences, which is being used to produce our molecular tree. And then we want to time calibrate this molecular tree. And we're doing this by adding the fossil data using morphological data. So we have to produce a morphological matrix of all the taxa that go into the, so the molecular taxa as well as for the fossil taxa. And then we have fossil occurrences, which you can largely just download from a paleobiology database, although that needs quite a lot of curation, so there's a bit of work involved with that. And then also abiotic factors. So, so the fossil occurrences and the molecular tree are then used to estimate speciation and extinction rates through time. And we will use these rates to address three main goals. So goal one is to look at possible trait-dependent increases in diversification. Are there any of these innovations that may have led to an increase in diversification? Are we seeing a diversity-dependent slowdown of diversification? So as species originate, more and more species originate, ecological niches are being filled, are we actually seeing a slowdown of diversification? And goal three would be to look at the effects of abiotic factors on diversification rates. Now, in terms of the molecular matrix, this is actually probably the most time-consuming part. And for this, we need fresh material for DNA sequencing. So we conducted various project expeditions, shown here with the pink circles. And we're fortunate enough to be in a well-connected community of bryozoologists. So we also had a lot of samples being donated to us from all over the world, given by the yellow dots. But what I would like to talk about is this expedition to the Svalbard archipelago, which was funded by Assemble Plus. And this is Piotr Kuklinski, who was my host. He works at IOPAN. And I have to say, he was the most magnificent host. Everything was extremely well organized in terms of the equipment, consumables, even dive gear. So I basically just had to turn up, which was brilliant. The boat operated 24 hours. So there are obviously the long-term monitoring measurements that they were taking and various other projects, including ours, that were accommodated into this schedule. Now the ship accommodation and labs is brilliant, really comfortable. The food is good, I should mention. That's not to be underestimated on a research cruise. So I can really recommend this facility if anyone is interested. So the expedition, we sampled 7 stations throughout this cruise, ranging from 50 to 200 meter depth. So we started off here in Longyearbyen, sampled a couple of stations in Isfjorden, then went down south to Hornsund, and then up to the north to Ny-Ålesund, another couple of stations, and coming back to Longyearbyen then. The equipment that we used was an epibenthic sledge, and this would be a typical catch. So it would be small rocks and shells would be encrusted by bryozoan colonies, which we would then identify under a stereo microscope, scrape off and preserve. A bit time consuming, but all worth the effort. So we collected 571 specimens from an astonishing 132 species of bryozoans. So I was really, really pleased with that. So this gives you some of the images of the species that we collected. So these are 20 species that we've already genome skimmed and coded for their morphology. And many of these only occur in the Arctic, so it was well worth going there. And in particular, we had on our hit list this species, Hinksipora spinulifera, because this family only occurs in the Arctic, and we didn't have any material for this family yet. So it was crucial to get material for this family. And then you also get some really quite interesting species. So here we have the Arctic species Harmeria scutulata, which forms subcircular colonies, which have in the middle normal feeding zooids. And then on the circumference, you get these miniature zooids, which are there for reproduction. So this is quite a unique way of reproducing. So in terms of the molecular phylogeny, we're not quite done yet. So we're still adding species to it, but we have a good phylogeny in place already. And I was going to cherry pick some of the things that we've already discovered from this phylogeny. So the thing to point out here is that the phylogeny is based on mitogenome data and 18S and 20S data. So we shotgun sequence the DNA and then skim off the genes that are then high copy numbers. So it's a genome skimming approach, so using Illumina sequencing. And the overall topology, there are some regions denoted here by the turquoise circles, which are difficult to resolve, but there are many parts of the tree which are nicely resolved. So in terms of some of the findings in this phylogeny, so with bryozoans, we have brooders and broadcast spawners. So we know that in this part of the tree, most of them are brooders, or all of them are brooders. And so they have these, I mentioned these already, the ovicells, these calcified brood chambers, which incubate the embryo, producing lessitotrophic, short-lived larvae. And then we have those that release fertilized eggs into the water, which then develop into planktotrophic larvae, longer-lived larvae. So that was always the status quo. However, as we were sampling this part of the tree, the supposed broadcast spawner group, more densely, we actually discovered a whole different range of brooders with very, very different types of brood incubation. I mean, I'm not going to go into the detail right now, partly because of time, but it's a bit like the licorice all sorts of brood incubation. So that was quite a surprise. And then we're also seeing some interesting results in terms of the frontal shield. So a bryozoan zooid, some of the early bryozoans that evolved, they didn't have a calcified frontal shield. So what you would get is this membranous frontal surface. And in some cases, you would have spines that are going all around this part, this membranous frontal wall. And from the fossil record, there were these hypotheses that these spines would overarch and form these cage-like structures, like here. So this is what's called a cribromorph frontal shield. So cribromorph here. And there was always this hypothesis that this type of frontal shield evolved from bryozoans like these. These are called caloporids. And we're actually finding that with our molecular data that often they are grouping together. So there is some truth in that. So we can confirm what we already know from the fossil record about the evolution of this type of frontal wall. And what's quite interesting is that you can see here the red dots on the phylogeny. These are the places where the cribromorph frontal shield pops up. So it evolved more than once. And similarly, we have it's a different type of frontal shield, the gymnocystidian frontal shield. So this is a fully calcified frontal wall. So here we have two back-to-back zooids. And what was quite interesting here is, again, they popped up in different parts of the tree. But this occurrence, so this is in the broadcast spawning clade. So another interesting result in this part of the tree, because most of the taxa in this part of the tree, they don't have any kind of frontal wall. So to find this type of frontal wall in that part of the tree was a complete and utter surprise. So it seems that bryozoans are extremely flexible in how they can form morphologies, quite drastically different morphologies. So once we have our molecular phylogeny complete, we're going to time calibrate it. And once you have a time calibrated phylogeny, so the branches are scaled to time, you can then produce a lineage through time plot, and the gradient of this plot can be used to infer speciation rates. To some extent also extinction rate, but less so, because molecular phylogenies typically don't include any fossils. So our approach is to incorporate the fossils into the phylogeny as terminals. So we have, so in red, so this is like a work, an intermediate time-calibrated phylogeny that we produced. So in red, we have our fossil taxa, which were included using the morphological matrix. So the nice thing about bryozoans, because they have such a massive fossil record, it's, we are spoiled for choice for our calibration points. And we have a good methodology now in place to produce this time-calibrated phylogeny once the full phylogeny is in place. Coming back to this slide, I just wanted to mention, so moving on from this part of the tree, the molecular phylogeny, we've also just submitted a paper to paleontology, which was a Bayesian analysis of the fossil record. So this is only using these ingredients, fossil occurrences, and the abiotic factors. So we generated origination and extinction rates based on just the fossil record. And in terms of our goals, so we visited those goals in that paper. Goal number 2, we are seeing some evidence for a density-dependent slowdown of diversification. And with regards to goal 3, we're seeing that we have a positive correlation of sea surface temperature and origination rate. We have a negative correlation of sea level and extinction rate, and a negative correlation of dissolved oxygen and extinction rate. So, so we're curious to find out what the results of the time-calibrated molecular phylogeny will give us. So something to look forward to. So that just leaves me to acknowledge a few people. So first of all, Assemble Plus, thank you very much for enabling me to collect all these Arctic species. The Leverhulme Trust, which was the overarching funder of Molecules Meet Fossils, of course, Iopan and Piotr Kuklinski in particular, the crew of the Oceania, everything worked extremely smoothly, brilliant. I mentioned already the food. They even let me in the kitchen on one occasion. Then I would like to mention my postdocs. So we've got Helen Jenkins, Paul Taylor, Farideh Moharek, and Rachel Graham. And we also have Mary Spencer-Jones, who's the Natural History Museum Bryozoa curator, and Andy Hall, who is a research assistant who's been making all the Illumina libraries. So that's a lot of work she was doing. So, and I should also mention, it's a very, this was a very international project involving most of the bryozoan communities. So lots of people provided specimens, logistics, permits. So a big thank you to everyone on this list. And thank you for joining this session. Thank you very much.
Thank you, Andrea. So if anyone would like to ask some questions, please indicate in the chat, and then I can, so this is more or less in a certain order. I have actually a question myself. So you're looking at the fossil record, and you mentioned there were many species, and you're trying to basically link this to the molecular phylogenies. So how plastic is the morphology of these species? Yeah. Because if they are, if they change with the environment, it may be that complicates a bit this integration into the molecular with the morphology.
Yes, so they are extremely plastic. In terms of within species, so we are seeing within species variation in some species. In terms of some of the, we can see it actually in zooid size depending on temperature, so that can vary. But for us, the biggest problem for us is, so the fossil record is immense, but there's so few bryozoan researchers working on them. So the taxonomy actually needs quite a lot of revision. So that's why, I mean, I'm working with fossil workers like Paul Taylor, who's the world authority on this, to help us with this, to actually, so when you're actually downloading fossil occurrences, for example, from paleobiology database, there's a huge amount of errors which needs to be curated. So the taxonomy needs to be reworked. And then we're also getting a lot of homoplasies. So when you are thinking about where to place the fossils, we are often in a bit of a conundrum where to restrain them. I mean, we are using phylogenies just with the morphology also to identify homoplasies, but it's a bit tricky.
It's a puzzle.
Yeah, but that's the fun of it.
That's why it's fun.
Yeah.
Okay, thank you. Anyone else wants to ask any questions? No. Well, thank you again, Andrea. It was a very nice talk, and well, good luck.
Thank you very much. Thank you.
Thank you. So we'll go to the next speaker. So the next speaker is Martina Copari, and she is a researcher currently, I think, at the Polytechnic University of March in Italy, and she was previously at the University of Genoa. And her interests are on coral biology. And during the Assemble Plus, she visited the Oceanographic Observatory of Banyuls of Sorbonne University, which had a presentation this morning talking a little bit about the marine stations in France. So Martina is going to talk about unveiling asexual reproductive traits in black corals, polyp bailout in Antipatella supinata. So Martina, when you are ready, you can present your PowerPoint.
Okay. Okay. Good afternoon to everybody. Thanks, Adelino, to introduce me, and thanks to give me this opportunity to present my results obtained thanks to the Assam Plus project, in which I participated in the 2018. My project was focused on the asexual reproduction traits in black corals, in particular on Antipatella supinata. Black corals are important components of temperate mesophotic ecosystems, which distributed in between 40 and 260 meter depth, where the photosynthetically active radiation is lower than the 1%. Black corals are also defined as ecosystem engineers because due to their three-dimensional and arborescent morphology, they increase the heterogeneity of the habitat, providing also food and refuge and nursery areas for many associated species, thus increasing the biodiversity. Despite the bright colors of black coral polyps, their name derives from the black and spiny skeleton, which is mainly composed by a scleroprotein named antipatin and also by chitin. This black skeleton is also exploited in jewelry. In the Mediterranean Sea, there are 4 main species of black corals or antipatarians, which are Antipathes dichotoma, Leiopathes glaberrima, Parantipathes larix, and Antipatella supinata. The bathymetrical range of distribution of this species is between 60 and 2,000 meter depth. Antipatella suppinnata is the most abundant black coral species in the Mediterranean Sea. It's also the shallower species with its bathymetrical range that range between 60 and 200 meter depth. And this species is also able to form extended and dense forest. Many aspects of the ecology of this species are already known, such as the geographical distribution, the growth rate, the diet, the associated microbiome, the symbiosis, and also the degree of vulnerability. But there are some other aspects related with the reproductive behavior that still need to be further explored. For instance, we know that this species reproduce sexually in summer, with the spawning event that occur at the end of August. We know that this species is a gonochoric species, but the spawning event has never been observed either in laboratory or in the field, and no information are available about the larvae of Antipatella suppinnata. In general, for black corals, there is a lack of knowledge about the larval ecology. There is only one previous study by Miller, which investigated and described for the first time the larvae of a congeneric species, which is Antipatella fjordensis. Moreover, we know that Antipatella suppinnata is able to reproduce asexually by fragmentation. But we don't know if this species is also able to use different asexual reproductive strategy. So we would like to investigate more in depth the reproductive behavior. That is why I apply for the ASAMPROS project, which give me the possibility to join the Observatoire Oceanologique of Bagnols-sur-Mer, which allow me to use the facilities such as the aquarium and the thermostatic rooms that I need to perform my observation and my experiment. But more important, I could join the research team with strong experience and knowledge on coral and larval ecology and coral maintenance. The sampling was performed on July 2018. 6 colonies of Antipatella suppinnata were collected by technical divers at 63 meter depth. While the divers were sampling, I was collecting water from the same depth in order to fill the 6 different tanks in which the colonies were placed for the transport. And the sampling location was Bordighera, that is on the west side of the Ligurian region. And then the colonies were transported to Bagnols-sur-Mer with a thermostatic van in order to maintain the temperature as constant as possible. The temperature at the sampling site was 14 degrees. And once in Bagnols, we placed the colony in a 2,000 liter tank, in which the temperature was controlled both by the thermostatic room in which the tank was placed, but also by the presence of a refrigerator. 2 pumps were positioned inside the tank in order to maintain the water movement and currents. And the colonies were fed twice a day by Artemia, Naupli, and Rotifera. The entire volume of water was exchanged in 24 hours, and prior the leaving of the thermostatic room, all this water was passed through a side tank that was equipped with a propagular strap. We positioned this trap in order to capture the fertilized gametes, the fertilized eggs, or potentially also the larvae. After 2 weeks of reading, we started to see some kind of stressful response from the colonies, because we started to observe that the polyps were often closed and the tissue became thin. After 1 month, we started also to observe the presence of many propagules inside the trap, which we identified as bailout propagules. The bailout is defined as an active detachment of the polyps from the mother colony as a response to stressful condition. The possibility of the polyps to detach from the mother colony allow the polyp in one end to survive and to the other to escape from the unfavorable condition. The phases that bring to the formation of the free-living propagules are summarized in this figure, and the process starts with the cenosarc withdrawal from the mother colony, followed by the detachment from the skeleton and the subsequent expulsion of the free-living propagules. We performed observation of these free-living propagules for 45 days. Every day we collected 10 propagules and we placed them in a small beaker filled with filtered seawater. And this sampling was done in order to observe if the behavior of the propagule changed during time, or if they changed their shape, or if they settle. What we observed was that most of the propagules survive, but any of the propagules settle. Since the bailout event was a massive phenomenon with many propagules produced every day, we sampled a subset of propagules of more than 2,000 propagules in order to describe the morphology and also to perform histological and SEM analysis. The main morphology that we recorded was the circular one, followed by the elongated and the polypoid shape. We hypothesized that the elongated shape derived from the detachment of the entire tentacle from the polyps, whereas the polypoid shape is the rearrangement of an entire polyp that detach from the colony. And the circular shape, instead, is the evolution of the previous morphology into the circular one. Regardless of the morphology, all the propagules were actively moving, rotating on multiple axes, and this capability of moving was due to the presence of cilia on the surface of the propagules. We also perform histological analysis, and in this picture you can see the section of a circular, an elongated, and a polypoid propagule. In the first picture, you see a complete rearrangement of the internal anatomy of the propagule, and there is still the presence of a thin layer of mesoglea indicated with M. There is the presence of an external and internal layer, and also many vacuoles of unknown function. In the elongated morphology, still present the enlarged base, which was the connection between the tentacle and the polyps. So this picture proved that the elongated shape was the entire tentacle that detached from the polyp. The polypoid shape still show the presence of a gastric cavity, as well as the septa that divide the one polyp from the other. On the surface, on the external layer, there is also the presence of nematocysts, which were identified as bicitric isorrhizas, the same nematocysts as we found in adult specimen, and also the same size. SEM analyses were also performed. In this image, you see a circular propagules, and on the surface there is the presence of many explosive nematocysts, and with B it is indicated the barb of the nematocyst, which is the spiny portion of the nematocyst. You see in one, with N indicated the capsule of the nematocyst, with B the spiny barb, and with F there is indicated the filament of the nematocyst. On the surface of the propagules, we found also the presence of many other filaments, much more thinner than the others, that were the cilia that allowed for movement. To conclude, the event of bailout was already described for black corals, but it was either mechanically induced by cutting a portion of the polyp, such as the tentacle, or triggered by stressful condition. What is interesting in our study is that we observed for the first time the spontaneous detachment of a portion of the polyp, the tentacles. And this particular phenomenon is similar to the pinitomy, which is an asexual reproduction strategy of some octocorals. Regarding the stressful condition that could have triggered the bailout event, in literature there are many factors are listed, such as the low food availability, the increase of temperature, an abrupt change in the oxygen level or salinity, and also a change in the pH. Since we checked all these parameters every day, we could exclude that in our case, the bailout was triggered by these factors. So we hypothesized that our colonies lately responds to transport and sampling, or maybe to rearing condition per se. Or maybe also the density in the tank was not appropriate, or the water movement. Indeed, many species are, some species are more sensitive than others of the presence of the aquarium. And after the 45 days of observation, we moved the mother colony to a big expositive tank after removing the necrotic portion of the colonies. And after 2 years, these colonies are still alive, they are healthy, and this observation has an important implication in natural environment, because if the factor that triggers the bailout is not permanent, then the colony have the potential to survive, to recover, and to survive. About the lack of settlement, we hypothesized that we did not observe settlement because our experiment lasts only 45 days. Indeed, in literature, some species need up to 7 months to settle. And also, maybe the glass beaker that we provide was not the perfect substrate to adhere and to settle. Another important observation is that the mechanisms that underlie the polyp bailout, such as the tissue and the polyp detachment, is similar to the mechanisms of the polyp expulsion, which is an asexual reproduction strategy. So it is likely that the Antipatella supinata that is able to bail out is also able to reproduce asexually by a polyp expulsion. And this has also implication in natural environment, because Antipatella supinata could reproduce asexually by fragmentation and polyp expulsion, and this could help maintaining the scattered patches where the sexual reproduction could not be effective. And finally, Since we saw that bailout propagules live for a long time, at least for 45 days, this could allow, this long-term survival could allow for a long dispersal under unfavorable conditions and the potential to colonize more favorable environment. And this capability might explain why we found a high degree of genetic connectivity among population in the Ligurian Sea. And with this, I concluded, and I thank you for your attention.
Thank you, Martina. So, are there any questions? I have a curiosity, mostly. Yes. Do you know what triggers reproduction in these corals? Is it temperature, light, and is there anything known about endogenous factors?
It could be related with temperature. Indeed, we know that normally in the Mediterranean Sea, the temperature rise from 14 to 16 degrees, and this could be the factor triggering the sexual reproduction. And I don't think it's light, because at mesophotic depth, the degree of light is not so the light that reach this mesophotic environment so low and is almost constant. So I think it's more related with temperature than with light. Mm-hmm. And it is probably that it's also some kind of chemical substances produced that help to synchronize the spawning, because in the end they are gonochoric species, so they need to be synchronized between male and females.
Mm-hmm. Okay. Any other questions? Well, in that case, thank you. So the next
thank you.
Talk will be in about 12 minutes. So we'll have a small interval because we have to keep to the time. Otherwise, people that may want to join don't, they would be finished by then. So we have to try to keep to the schedule. And I know that the next speaker has some difficulties in joining. So let's hope he can do it. So see you soon.
First-class scientific research. First-class scientific research relies on effective, convenient access to tools, facilities, and data. Assemble Plus is a European Union-funded research and innovation program with a consortium of over 20 partners that integrates key marine research facilities across Europe and beyond, offering access to top-tier research infrastructure through a competitive application procedure, evaluated on the basis of a feasibility assessment and research excellence. Whether from academia, industry, or policy, through its easy and straightforward application process, Assemble Plus provides scientists with on-site or remote access to biological resources, varied ecosystems, experimental facilities, technology platforms, e-infrastructure, and expertise, and provides lodging and catering support over the course of their placement. Assemble Plus also performs its own networking and research activities, ranging from interacting with new users and businesses to cryobanking marine organisms to providing diving services for researchers. Over the course of the project, as well as providing access, Assemble Plus aims to strengthen transnational and multidisciplinary networks, create public-private partnerships, enable new technologies and services, upskill researchers, and improve the long-term sustainability of Europe's marine biological stations. So, if you're a researcher in need of access to marine infrastructure, such as laboratories, equipment, or any other provision, Assemble Plus welcomes proposals for access on a rolling basis from the 29th of August 2018 to the 30th of October 2020. For more information about the project and call for access, please visit www.assembleplus.eu.
Well, welcome back. So I think Danilo Cagliari is now managed to join us. He's going to be the next speaker. He's associate professor at the University of La República in Uruguay, and he's a marine ecologist interested in plankton that visited the Christineberg Marine Station at the University of Gothenburg. And he's going to talk about a straight story, riding the microzooplankton from South America to Northern Europe and back. So Danilo, I hope you can make a presentation now. I saw him in the audience. His name was there. Ah, okay.
Hello, Adelino. I'm trying to just start my video now.
That's fine.
Here it is, and now I will try to share my screen. Can you see that, my presentation?
Not yet. It's probably taking some time. It is now. We can see it.
Okay, thank you. So thank you very much, and good morning to everyone. As it was presented, I will give this talk on the results of an experience, a visit to Christineberg in Sweden, where we developed a very specific and simple project on marine microzooplankton. see. Okay. Just to get started, microzooplankton is that very heterogeneous collection of organisms which have a few key characteristics in common. That is, first, they are planktonic. Then they are small in size. They are between roughly 2 and 200 microns approximately, and they are mostly protozoans, that is single-cell organisms, which that is mostly ciliates, flagellates, plus a few metazoans, mostly planktonic larvae. And another important characteristic they have in common is that they are either heterotrophic or at least mixotrophic. That is, that they can develop both heterotrophic and autotrophic metabolisms at the same time. These are very important. That's a very important community because they are currently considered to be the main herbivorous organisms in the ocean. They probably consume in the order of 2/3 of the global oceanic primary production. Due to their small size and rapid generation times, they are able to control phytoplankton population dynamics. That is, they have very microzooplankton and phytoplankton have tightly coupled population dynamics, and due to their impact on primary production, they are also key players in biogeochemical cycles in the ocean, because the size of the largest predators within the microzooplankton are large enough to be part of the prey of other larger plankton organisms. They constitute also a very important functional link between classical and herbivorous microbial webs. A lot or much, perhaps most of what we know about the role of microzooplankton and their herbivorous action in the ocean is derived from what is called the seawater dilution method or technique that was first introduced by Landry and Hassett in the early 80s. This is a classic contribution that has been very highly cited and used, and it's very popular because it is based on a very simple rationale and involves very simple logistics. That is, basically one needs to take a natural sample of the microplankton community and perform a series of dilutions. The key point here is that as we dilute the sample, the encounter rate between predators and prey decrease linearly with the increasing dilution. And so this dilution, the effect is that it weakens that tight coupling that exists normally between the microzooplankton predators and their prey. That allows for an increase in the population growth rate of the phytoplankton prey. Okay, that is normally the phytoplankton population dynamics is modeled as an exponential process where we have the key variables that are K, that's the intrinsic phytoplankton growth rate, D is dilution, and She is the grazing mortality of the phytoplankton. This equation is straightforward to linearize, and then we have a term on the left that represents what is called the apparent or net population growth. Then we have the intrinsic growth rate, that is the intercept of the line, and the grazing rate by the microzooplankton is represented by the slope of the line. It looks more or less like this, a typical result of a dilution experiment, if one is lucky enough, of course, where we have the intercept and the grazing mortality as the slope Okay, there are, of course, as always, very important two main, say, broad assumptions behind this method. First of all is that the phytoplankton specific growth rate is constant and across dilutions. That means that all factors important to modulate the phytoplankton growth should be equal between all the dilution conditions. That means mostly light and nutrients. Nutrients is what is usually considered that is potentially problematic. So the typical solution to this that has been discussed quite a lot in the literature is to add some small amount of nutrients to ensure that there is no preferential limitation in, say, more concentrated treatments. And the second big assumption that is more problematic is that per capita grazing rate of the microzooplankton is linearly related to dilution, that is to the actual encounter rate between predator and prey. As I said, this is quite more problematic and has been dealt a lot with in the literature. The key issue here is that first of all, there are non-linear feeding dynamics because the microzooplankton has its own or their own functional responses to food, and then there is an interaction between these functional responses and the actual absolute prey concentration in the dilution series, which are different among conditions. And then one is quite common that we observe prey concentrations that are above saturation levels in the feeding behavior of the microzooplankton at low dilution conditions, or we may find that prey concentrations are below the feeding threshold at highly diluted conditions. This creates strong departures from the linear pattern that we saw just before. And these issues have received a lot of attention as well, and there are several ways to get around them by adapting either the experimental design or the data analysis. But there is still more to per capita grazing rates, and there are some issues that have not been still properly addressed in the literature. The key point here is that dilution implies a change in the concentration of prey, but also of predators. And so the immediate question, or a kind of obvious question, is what happens to eventual density-dependent effects of microzooplankton ingestion rates? By density dependence, of course, we understand that phenomenon where the performance of a given function or process progressively changes with the concentration, that is the density of individuals within a population. Normally, people understand that there could be positive and negative density-dependent effects. The positive are those that when the performance of the function increases or gets better with more density, and that's a typical case of, for instance, the spreading of contagious diseases. And then the negative effects that are the most common is when the function decreases its value with increasing density in the population. These density dependence effects are, I mean, in the ecological literature, are very well known, and it's well established that they are general, that is, they affect different kind of processes. Specifically, I mean, it's easiest to think about that, say, physiological or metabolic processes that are, and the effect is primarily at the individual level, but it cascades at population and community levels as well. And it's pervasive in the sense that it affects basically populations in the whole tree of life, say from bacteria or viruses to mammals. However, there is, as I said before, they have not been evaluated in the microzooplankton, and even less in the context of dilution experiments. And there is no reason to think that the microzooplankton would be extent of this kind of phenomenon. So the general working hypothesis that we worked for this project is that negative density dependent mechanisms operate and shape ingestion rates in marine microzooplankton. And that's what I will show next. If this hypothesis is valid, then this is the kind of pattern that we may expect to find. Say, if we plot the consumption rate of the microzooplankton assemblage versus the density of the microzooplankton population, we should find what is described here by the continuous line, a positive response, but with non-linear, with some evidence of saturation. And if we look at the per capita ingestion rate of the microzooplankton, then we should find a negative slope that could be linear or with some other shape, but there should be a decrease in the per capita ingestion rate with increasing density. Okay, so as stated, we worked in this project in the Kristineberg Marine Research Station on the west coast of Sweden. What we did was a modified version of a dilution experiment. We took a natural community from the Gullmar Fjord, that is the sea area next to the station, and we performed a manipulation in which, by double serial reverse filtration, we concentrated, we got the predators in that sample, all those predators that were between 10 and 200 micrometers in size. And then we resuspended and diluted that community of predators in 5 levels with densities between 0 and 50 individuals per milliliter of the microzooplankton. And to them, we added a cultured alga, that's Isochrysis galvana, at a known and constant amount per in every treatment. That amount was 60 micrograms of carbon per liter. So we had in this way we had a design in which we had a dilution series of predators, but constant phytoplankton food levels across conditions. All other conditions were more or less standard. That is, we worked in relatively small bottles, 250 milliliters. We incubate them for 24 hours under a light-dark regime and temperature that matched more or less the in situ conditions at that time of the year in the Gullmar Fjord. We added nutrients as recommended. We counted we estimated the biomass of phytoplankton at initial and final times by measuring cell size and cell numbers with a culture analyzer, and also using chlorophyll A as a tracer. And we counted the microzooplankton by inverted microscope. And then I go straight to the results. This is what we found in the natural microzooplankton community. We say more or less a rough or proximal composition of the community. We see that there was a overall high abundance of microzooplankton in the order of 14 individuals per milliliter, and that community was strongly dominated by dinoflagellates in the first place and ciliates on second place. That is more or less what could be expected. And here we see some of the patterns that we observed in the experiment. This is the algal net growth rate plotted against microzooplankton density. We see a strong decline in phytoplankton growth as we move from highly diluted to more diluted to less diluted predators. Say that's more or less what it would be expected. The more predators, the more they eat, and the less the net growth of the phytoplankton. And then here is what looks like the community grazing pattern, that is grazing against plotted versus microzooplankton density. We see that there is a positive trend, but that trend is non-linear and shows some sort of saturation effect. We didn't arrive to the asymptote. We didn't see the maximum grazing rate, but the signal is clear. And finally, the per capita grazing rate, in this case, it clearly decreases rather strongly with increasing microzooplankton density. This is the set of results based on algal cell count and size measurements. And just very briefly, I show the equivalent results, but based on chlorophyll A as a tracer of phytoplankton biomass. Basically, the patterns are very consistent, and there are small differences, of course, but the information is basically the same in one set or another set. So our interpretation from these results is that in the microzooplankton community and per capita grazing rates were consistent with the presence of active densodependency affecting ingestion rates at ecologically relevant levels of microzooplankton densities. These densities that we work with are, that's important to note, they are within the range of naturally observed abundances or densities. These effects, densodependency effects, were clear at high and even medium densities, and we interpret that the arising of this effect has to do with some kind of allelopathic or interference effect associated to crowding. That is, however, the ultimate mechanism is, of course, unknown. The microzooplankton is composed of very different, but multiple very different taxa in terms of their biology and ecology, and their feeding modes, etc. So there probably is not a unique mechanism, but a combination of several that give this aggregate pattern that we saw in our experiment. So the key implication that has this result is that some of the assumptions of the seawater dilution method may not be valid if density dependence is significant at in situ conditions. The overall effect that this would have is that density dependence would flatten the slope of the regression line. That is, they would of the regression line in a typical dilution experiment. That is, they would lead to an underestimation of real grazing rates. Finally, I would like to stress that this is a first experiment that shows this point. And of course, it would be very important to have further results of similar kind in order to be able to generalize or revise these conclusions. Finally, in the last minute, I would like to say a final message. What I just told is a straight story, but not the real story. The real story is more complicated. The thing is, I must say, my original research plan here in this visit to Christiansø was within a 1-month schedule. I had planned to run some experiments with another scope. the idea was to perform experiments with size fractionated assemblages in order to identify regulations within the microzooplankton assemblage. But the point is that in the first 2 weeks, we performed many pilot experiments, and we got very weird patterns in the data, which are, of course, in the end, not uncommon, but they are not always reported in the literature. And where we saw like real flat, no grazing activity, or even some sort of increasing growth rate of the phytoplankton in less diluted conditions, which is the opposite pattern of what would be expected in a dilution experiment. So there was some kind of apparently negative process that was in action and compensating for dilution. And we were like 15 days looking for what that could be. And in the end, by kind of some kind of intuitive reasoning, we thought that maybe this old pattern could be related to, as I say, some kind of negative process that could decrease the intensity of feeding when more macrozooplankton were present. What that could be? Well, densodependency was an obvious candidate. So my last words here are that it's a sort of evaluation of my experience in Assemble Plus, is that if positive results were in the end viable to get from this, is that for at least 3 important features of the instrument. First, which is the most obvious, of course, is the access to instruments, laboratories, logistics, and so on. That's very well known. But I wanted to highlight the flexibility in terms of the possibility that the researcher has to find the time to explore, fine-tune the experiment, adapt the experimental setup, and even, as it was the case here, change the research plan altogether if that is necessary. And that, I think that's very important. And last, but maybe the most important of all, is the access to communicate and directly interact with colleagues with whom share thoughts and plan alternatives, discuss ideas that allow the original research plan to move forward. So thank you very much.
Thank you, Danilo. So does anyone have questions? Please say in the chat that you have a question, and then you can then ask the question verbally. I can start by, I'm completely ignorant in this kind of subject. So if you were to do these experiments at different latitudes, would you expect similar results? Or is this a general feature of the microphytoplankton?
Thank you. Well, actually, yes. As long as the densities of the microzooplankton are high enough to trigger densodependency behaviors, yes, I would expect that to be universal.
Okay. Still haven't seen any questions from the audience. Okay, Danilo, thank you very much for your effort with all the initial difficulties in getting in. But thank you very much. It was very interesting, and I'm glad that it worked. So now we move on to the next speaker. That will be James Woggett. He's from Bangor University. He's a lecturer at Bangor University, and his research focuses on understanding how behavioral and environmental factors influence the distribution of cetaceans and seabirds in space and time. And in Plus, he visited Imar in the Azores, so in the islands that are halfway between Europe and the US, in the Middle Atlantic part, and so they are also part of the MBRC, the Portuguese node of MBRC. So, James, please, you can start your presentation.
Okay, checking everyone can hear me. Okay.
Yep.
Yeah, all good. And just share my screen. Can someone just let me know how to share my screen? That's okay. So let's have a look. I'll share screen. Yep, got it. Yep. Okay. Just checking people can see my. Can people see my PowerPoint screen now?
Yep.
All good. Yep. Okay. I'll go into full screen mode. Okay. Yeah. So sorry, could you just confirm you can see the whole screen now? So the first slide.
It's working.
Brilliant, thank you. Yeah, so yeah, thanks for introducing me. So yeah, I'm a lecturer in marine biology at Bangor University, and I'm going to talk about some experiences with the Assemble Plus program, which I first used to go to Vigo, so in Galicia in Spain. And what I'd like to show here is actually how what I experienced and what I've achieved as part of this program has really steered my current research. So I'm doing things now which I wouldn't necessarily have done before, and I think it's been a great opportunity to study different systems, interact with different people, and really sort of shape my research for the next few years. So to state what everyone knows, we're all very aware that environmental change is a big topic at the moment. And even though temperature increases probably get the most attention, we also know that our patterns of weather are changing quite a fair bit as well. So in particular, we're seeing changes in precipitation, so rainfall and wind. We're seeing changes in the intensity of these two factors, so perhaps wind speed or the amount of rain, but we're also seeing changes in the frequency, so how often we get storms or how often we get periods of wet or windy conditions. And we also see geographical variation. So these are just some examples that I've taken from the literature, of which there are many examples, just showing that the impacts of change in weather is not necessarily the same in all places. So some places could get drier, some places could get wetter, some places could get calmer, and some places could get stormier. So there's going to be different impacts of changing weather patterns dependent on exactly where you are. In terms of impacts on seabirds, which is the focus of this talk, we do know that extreme weather conditions can have quite bad impacts on seabird populations. So these are birds which spend most of their time feeding at sea in coastal waters, and we know that very prolonged storms can constrain their movement and their activities. So birds could seek shelter either in sheltered bays, or perhaps they will spend more time roosting on land next to the coastline. And by doing this, obviously, there's less time to feed. So these very long storms can prevent birds feeding. And in the case of some birds as well, so I've picked an example here of a European shag. Persistent cold winds can also cause hypothermia. So these birds aren't completely waterproof, which means that their feathers get quite wet. And when they get wet and when you get cold winds, they're quite susceptible to hypothermia. And what this causes is in periods of bad weather, you get what's called seabird wrecks. These are where you get large numbers of birds, a high amount of bird mortality, with lots of birds found dead on beaches. So these seabird wrecks can have population level impact. So this is some work done a few years ago now, and it's showing that in periods of very intense storms, the likelihood of an adult surviving the winter is really low. So in short, extreme storms are bad for seabirds. But what I'm interested in here is non-extreme weather. So we're all very aware, and I think it's quite obvious that bad storms have bad impacts. But what about these day-to-day variations in wind and rain? So they're not particularly wet, they're not particularly strong, but obviously they are, it's slightly wetter or slightly stronger. And birds need to cope with these daily variations in order to successfully find food. But what we do know is quite a lot of seabirds have very strong foraging routines, which is associated with topography and tide. So in coastal waters, obviously tides are a very prominent feature in most places where you get increases in tidal height, sorry, in depth, and decreases in depth, but also changes in the speed and direction of flows. And we know that these can create very predictable feeding locations, which cause these routine feeding behaviors in birds. This is some work that I did a few years ago now, which is quite a good example of this, where we found terns were feeding on these shear lines. So these features you see in the bottom left, very predictably at certain times. In this case, they were feeding on these shear lines when they emerged during the flood tide. But what I want to know is how do the changes in wind speed and wind direction, in rainfall, change the way these birds use these features, and can that have population level impact? So can consistent disturbance to these routines actually have population level consequences? So some examples of how these things can potentially impact seabirds' feeding behaviours. So say you're a seabird and you feed it on that area in the north, so the red star. You could have a plume which typically is constrained to the mouth of the river. You have intense rainfall, that plume gets bigger, and perhaps you've got fish which are feeding in that plume on the nutrients brought down. You then get northern winds that could force that plume into your area, so you could perhaps benefit from prey. But if it goes the other way, you can't. So this is a potential consequence of that interaction between wind and rainfall. When it's calm and dry, sediment will tend to settle on the seabed. No additional sediment is added, and the water stays clear. But when you've got a period of wet and windy conditions, sediment gets resuspended. Sediment is added from the groundwater runoff, and the water becomes murky. So say you're a bird which needs to see its prey to feed, that could be important. And again, for some birds where they're traveling long distances to feed, when it's calm, so you've got low winds, it's quite easy to fly, and it's quite easy to dive through the water column in search of prey. But then when you get wind, obviously it becomes a lot harder to fly, because you might be flying into winds. That water becomes turbulent, becomes mixed, and it's quite hard to dive through the water column. So potentially there you've got problems to feed if it's windy. This is the question which our research as part of the Assemble Plus program has sought to ask. So how do non-extreme weather events impact seabirds? I'm going to focus on 4 things here. So reduced visibility from precipitation and wind, perhaps nutrient enrichment or the addition of prey which are brought in with estuarine plumes, increased foraging costs from wind, but also to try and see how important these different processes are. So we know that tides are really important for birds feeding in the coastal areas, but how important is tide compared to wind and rain, for instance? So this was all, the first part of this project was performed in the Rio de Vigo, in Vigo in Spain, and we were hosted by ESIMAT at the university there, and they were fantastic hosts, and the several times we've been there, they've been really good to work with. So if they are listening, I'd like to thank everyone there for their help during our work, because I think we've hopefully got some really interesting results coming out of this. But just so people who don't know where this is, so this is in northwestern Spain. It's a semi-enclosed ria, so a estuary in itself. And we focused our surveys on the outer part of that estuary next to the Îles de Sèze. There is mild tidal currents, so there are some flows there, but they're not particularly strong. The main influences on this site is the weather. So it's very wind influenced, so it's a upwelling system. So the direction and speed of the wind can influence the amount of nutrients come into the system, and also the exchange of warm and cool water masses. And at times of the year it gets lots of rain, and this can influence the strength of the plume, which it's the Western Iberian buoyant plume, and it emerges from the Minho estuary, which you can see in the central map. And then during northern winds, that plume is pushed up into the ria. So at times that water in there can be quite brackish, so it can be quite low salinity. And what we did quite simply is we went out in the boat and did lots of transects, going forwards and backwards in that site, with the aim of recording the numbers of birds there. And we obtained environmental data on the salinity and the tidal currents from simulation models. And the idea then was to simply compare the numbers of birds with the environmental conditions in that site. And this is just some outputs from a IBI oceanographic model showing salinity. And you'll see this is the days in which we performed our seabird surveys in the outer ria. And what you'll see is on the first day, so on the 6th of the 6th in 2018, there was a noticeable plume emerging from the Minho estuary in the south. So this is when we had the most precipitation. But because the main river was to the south of the study site, this is where we saw the most freshwater input. But then, as you can see, over the course of the week, as the winds forced that plume to the north, it's entered our study site around the 10th and the 11th of June, so about 4 to 5 days later. So this plume gradually worked its way into our study site. And interestingly, when you looked at the counts of seabirds, so the counts of the seabirds there are on the bottom 2 plots. So you'll see European shag in the 3rd plot down, and ear-led gulls in the 4th plot down. This was when we got our peak seabird counts. So we saw more seabirds feeding when that plume had entered the outer ria. And there was some more gulls during the latter stages of our study period, during higher winds. But I think the main story here is when that plume entered the site, we had a huge increase in the numbers of feeding birds. And that was our conclusion from the first part of this work. So increases in foraging seabirds occurred several days after high rainfall when the plume entered the study site. And there's various reasons why we think this could be the case. It could be that we know that the European shag in that site sometimes feed quite intensively on sand smelt, which is a fish species which is associated with brackish water. And it could be that the plume brings with it big schools of sand smelt. But obviously this is all speculation at the time. The main story here is that they increased in number when the plume arrived. So what we did then, still in the Ria de Vigo, is we went back in and we looked at one site in a bit more detail. And this time, rather than having simulation models, we actually collected some empirical data ourselves. So you'll see here in sort of red is the transit route. And as before, we went forwards and backwards, counting birds over different days and over different conditions. But this time we measured salinity, temperature and turbidity with a CTD at the point labeled D there. And there's also a weather station next to that site on the Northern Island. So this time we've got concurrent empirical information on numbers of feeding seabirds, turbidity, salinity and temperature, but also the fine scale weather conditions, so wind and precipitation. And just to look at this plot now, so this is all that information combined onto one plot. It's a bit complicated, so I'll just take you through it slowly. The grey bars are the numbers of feeding seabirds in that site. The green is the turbidity, so how murky the water is, and the blue is the rainfall. So what you'll see there is on days immediately after heavy rainfall, there tends to be an increase in turbidity. So there's a reduced visibility, and the numbers of seabirds drop substantially. So I have standardized the scale there just so we can compare different components. But the peak seabird counts on the highest days were about 200 birds. So you'll see there that we're going from around 150 to 200 birds on some days, and on days immediately after increased rain, that drops down to say 5 or 10. So substantial decreases in birds on days immediately after rainfall. And we think this is because the increased turbidity just means the birds can't find fish. So these are sight feeding birds, and they're not quite finding fish as easy. And just to show you that a bit more simply, in the bottom left, you'll see a simple comparison between the numbers of feeding shags and the turbidity, and you'll see there that around sort of 1.5 TBU, the amount of birds drops off. So there seems to be a threshold. Interestingly, however, you'll see that 5 days into a prolonged rainfall event, so after 5 days of continuous rain, turbidity seems to stay high, but the birds come back. So is this that plume entering the area after 5 days? We're not sure, but that's just an interesting side result. I think here the main result is that after rainfall, increased turbidity, there's less birds. So just to add to our conclusions, we'll see that there's substantially fewer foraging birds immediately after high rainfall associated with high turbidity. But if we take these two results in combination, it seems to show a scale-dependent process. So short-term responses to high rainfall because of increased turbidity, but in the long term, when that plume arrives, the birds start to feed again. So similar to the previous talk, I think it's more complicated than we're trying to portray, but there does seem to be this interesting relationship depending on the timing of the rain. So just for the last sort of 5-10 minutes, or 5 minutes rather, I'm going to go into what this has led us to do post our trip to Vigo. So this is where I work. This is a lovely part of the world in North Wales in the UK called Menai Bridge. My office, when I can get to it, obviously not easy at the present time, but is on the left-hand side just by the big bridge, and I live just to the right. So this really is a local study site. And the idea here was to take what we learned in Vigo and try and apply it onto a place where we can do long-term observation. So the longer we can observe birds, the broader range of weather conditions we can cover, and we can perhaps start to really tease apart the various influences on their feeding behavior. So hopefully you'll see a working video. Apologies if not, but the thing about the Menai Strait, which is different to Vigo, is you've got a huge tidal range and very strong currents. So you'll see here, this is just some camera trap footage, and you'll see here that there's a huge range in the height of the water over the tidal cycle. So at its most, the water depth can be 7 meters higher than it is at, 7 meters higher at high tide than it is at low tide. And there's very strong current speeds as well. There's multiple freshwater inputs. So unlike Vigo, where there's one big river, there's lots of small rivers entering this system. It's quite windswept, so we get winds coming in off the Irish Sea. And as anyone who's lived in Wales before knows, it's very wet, so we get a lot of rainfall here. So basically there's lots going on, strong tides and lots of impacts of wind and rain. And what we've been doing for in sort of past half year is doing lots of shore observations when we can, obviously, depending on current restrictions. But we've done lots of shore observations at these four points along the strait. We've got CTDs in the outer strait that have been there for several months now, and we've got a weather station as well. So similar to the setup in Vigo, but these are all looking at a much longer time period. So we're able to cover a much longer time period because obviously this is our local area. And the main river input here is in the central strait, so that's where we have our local freshwater input. You've got very strong currents, but quite sheltered in the south, and quite exposed and slower currents in the north. So basically a good setup. And I can't really go into the final results in detail here because they're still ongoing. But what I can show you now is these are some of the counts from the various sites. And you'll see there that in some sites, so at Penmon at the top, which is the most northerly site, there is definitely a cyclical pattern. And this is because the birds tend to feed there more at low tide. So we've gone to these points every morning. So obviously over a period of time, you get to cover all tidal cycles. And you'll see there that there's certainly a sort of cyclical pattern. And birds tend to feed at Penmon and also at Menai Bridge at low tide. So perhaps when it's easiest to reach the seabed, because these birds feed on demersal fish. But in the middle region, so at Gallows Point, there's a bit of a pattern, but it's a bit more sporadic. It's not as clear. And we think that this is potentially the influence of the freshwater input in those middle regions. And again, just the second last slide, I'm starting to sort of develop models now that takes all these things into account, and we can try and simulate birds' movement around the strait. So obviously we can't do that at night, so that's where there's obviously big gaps in coverage. So we're going through night time now, so you can't see anything obviously, but during the day, we're managing to simulate birds' movement around the strait in response to short-term rain and short-term wind, long-term rain and wind, tides, and salinity, which is the input of fresh water into that system. But this is obviously work in progress. So not many conclusions here, but tides here seem important. So a lot more important than they did in Vigo. But still, there seems to be declines in feeding birds during strong winds and after high rainfall, and then increases when that freshwater plume enters. So again, this scale dependency, where you have these short-term negative responses to high rainfall, but then longer-term positive when that plume arrives, seem to be present in this system as well. And also something that we've also seen is we're seeing increases in birds in sheltered areas after prolonged high winds, but that's I don't think there's anything new there really. But yeah, that's just the final bit. So lots going on at the moment, and unfortunately I can't present more final results, but that's the end product of my work in Vigo. So it really has kind of made me look into topics that I haven't considered before. It's made me extend my work into different areas and ask different questions, and in that respect it's been very useful. So yeah, just what next? We're going to continue field work in the strait. We've got some GPS tagging of birds, look at their movements to quantify their routines and how they respond to different conditions. Developing camera traps, so time lapse to try and monitor, to provide continuous monitoring bird behaviour. And that model I showed you a few slides ago, we're going to try and get that even better. And once we've got that, we can start exploring with climate change scenarios, but also perhaps scenarios in land use. So yeah, just lots of people to thank, and I realise I'm tight on time, but yeah, I'd like to thank everyone involved, but particularly those at Invigo. So hopefully I've included everyone of relevance there, and thank you very much.
Thank you, James. So if anyone has questions, please type in the chat so that we know who wants to ask the question. I have a quick question. So these birds, they basically go away when there are, or don't appear when there are some not so favorable conditions. So where are they going?
So the birds that we studied, so the European shag and the gulls, these birds will tend to roost on land. So when they're not at sea, they'll typically be roosting on cliffs or rock ledges, and basically trying to sort of shelter from the storm. So it would be interesting to actually look at birds where that's not a choice. So perhaps shearwaters or puffin type birds don't have that choice, so they might have to sit out the storm at sea. But with these birds where they basically sleep on land, so the shags and the gulls, when they're not at sea, they'll be on some sheltered cliff or rock, or perhaps even a beach. Yeah.
Okay. Thank you. Anyone else? No. Okay. Thank you very much, James.
Thank you.
Also, I would like to thank all the speakers and all those that were attending these lectures. So tomorrow we start at 9:30 European Central Time with a keynote lecture by Esther Serrão, and I hope you can attend those lectures. And of course, the program will continue during the day as well, with some presentations from marine stations and further presentations from assembled visitors. Well, thank you very much, all of you. I just remind you one thing is that, okay, there are different ways of people connecting each other, but the platform, the B2Match, allows people to kind of book interviews with each other if you want to do that. So again, thank you very much and hope to see you tomorrow.
At NHBS, our purpose is to support those who are passionate about wildlife, ecology, and conservation. We stock a variety of books and equipment to suit the needs of marine conservation professionals, and our innovation and research team are here to help develop custom products for any project. We are happy to provide advice and to support you before, during, and after your purchase. Visit nhbs.com today to find out more.